Hello, everyone. In this video, we will explore the concept of fear, as well as how it is learned, and we will look at a classic experiment to apply it. But first, let us discuss what is fear. So here are some definitions provided by Merriam-Webster and the National Institute of Mental Health. But fear is an unpleasant and often strong emotion caused by the anticipation or awareness of danger. And it can also be thought of as the activation of the brain's defensive motivational system to promote behaviors that protect the organism from perceived danger. So to better think about that, we can really look at why do we experience fear? So especially if it gives us an unpleasant feeling. So fear really is designed to protect us. For example, fear of heights protects us from carelessly falling and hurting ourselves. And additionally, fear allows us to learn what to avoid and therefore can be a valuable learning tool. An example of this would be touching a hot stove and feeling pain immediately afterwards. We learn to fear touching a hot stove or a hot object in order to avoid the feeling of pain and getting burned. So we have all experienced the feelings of fear and different types of fear. Now, these fears can range from being scared of snakes or being afraid of heights, as we discussed in the last slide, but both are ultimately due to the fear of death. Now, what we want to look at now is fear an emotion that we instinctively possess, or is it learned? And what's interesting is fear is actually of two types. So there is innate fear that we do possess, and there's also fear that is learned. Now, innate fear is not well understood in mammals and is still being researched presently. So, for the topic of this lecture, we will only focus on learned fear. Now, one important point to note is that learning of such fears occurs subconsciously. So, for example, when a kid is trying to learn math, so he's trying to focus on what 2 times 3 is, and he learns that it's 6, he or she is aware of this learning. So it's conscious learning. Now, with fear, it ends up being subconscious learning. So it is something that is not being intentionally learned, at least not consciously intentionally learned. So in the previous slide, we considered the fear of touching a hot stove. And uh, when we do not know a hot stove would cause pain, we would touch it without fear the first time. However, when we experience the pain after the first touch and that sensation caused by temperature and pain sensors in our fingers ends up being sent through a neural pathway that connects it to the brain. And after touching the hot stove for the first time and experiencing that pain that's sent through those signals, our brain learns to associate the hot stove with pain, and therefore we learn to fear touching hot stoves. Now, what changes in the neural pathways in our nervous system and brain that's responsible for the learning of that fear? So that's really what neuroscientists have been trying to investigate um, and look at the neural pathways that implement this associative learning, which we will first explain in our next slide using Pavlov's famous dog experiment. So with Pavlov's conditioning experiment, there's a little video here that you can click on if you want to see it, and I'll quickly talk through what he looked at. But Pavlov designed a really good method to find out how associative learning works and how learning to unconditional stimuli works. So, for example, he had dogs that had food presented to it. Now, typically, when a dog is presented with food, dogs salivate, and this is an unconditioned response. So, the unconditioned stimulus is the food, and the unconditioned response is the salivation, which means it's not an intentional response by dogs. They're just hungry and they're looking forward to eating the food. Now, what Pavlov did is he paired this with a conditioned stimulus. And in this case, it was bell ringing. So if a dog hears a bell ring by itself, the dog will not respond or do anything. Now, what Pavlov did was Pavlov paired both the unconditioned stimulus, the food, and the conditioned stimulus, the bell, together. So when the food was presented to the dog and the dog salivated, Pavlov would also ring a bell. And what Pavlov showed was that after associating and ringing the bell with the food, and the dog, after doing this a couple times with the dog learned that the bell was associated with the food. And then when the bell was rung by itself without the food, the dog salivated. So what we're learning, and in this case, that is an unconscious response, an unconscious learning. And so what we're focusing on in this experiment is how that applies to fear and what neural pathways are are involved. So the Pavlovian auditory fear conditioning. So this is an experiment used to train rats to fear a particular sound. So first you have to know that when a rat is fearful, it will either freeze or run. So in this experiment, rats are in their cage. So rats are in cages so they cannot run. So in this case, fear is exhibited by freezing. And the level of fear is scored by noting how long a rat freezes. And there are automated programs that are available to score this using the experiment video that we can take a look at in just a little bit. Now, different rodents exhibit different levels of fear, as same with humans. But what the rat fear conditioning experiment looks like is, and this is similar to Pavlovian conditioning, except this time we're pairing tone with shock on the rats and then observing to see how they learn fear. So first what happens is rats will first hear a tone by itself and then get habituated to it. So rats will hear a tone by itself, and after a while the rats will learn that the tone doesn't really mean anything. So that's what habituation is. They get used to it and they ignore it after some time. And then in the next stage, the rat then hears the same tone, but this time it's accompanied by a little foot shock, causing it to feel some pain. So this is called a conditioning trial because the shock is being conditioned with the tone. And the number of conditioning trials typically varies from 5 to 10, depending on the intensity of the shock. But what ultimately happens is after the conditioning trials, the rat will then hear the tone by itself and it will have a fear response. And this fear response is scored because the rat has learned that the tone is associated with the shock. Now, this is similar to dogs learning that the bell is associated with the food and they salivate. But in this case, the rats end up exhibiting a freezing response. So with this, we have a video devised by Gregory Quirk's lab that shows the experiment that we just described. And I want you to go ahead and click the link and watch it. And while you're watching it, try to think about how the rat would respond after conditioning and how the rat would respond when it hears the tone before the shock, before the conditioning trials, as well as after. And you can go ahead and pause the video now and click on this Quirk video link and go ahead and watch it. Okay, so now that you're back and have hopefully watched the video, can you guess why the rats behave differently after conditioning? So what neural pathways may have changed and how? So take 2 to 3 minutes to jot down your thoughts, pause the video again, and think about what happened and what could have been going on in the brain that caused this change. Okay, so let's recap this experiment in fear conditioning. So in our experiment, the rats appeared to initially not respond to the tone, as we saw by that habituation process. And at the beginning, they continued their behavior without any type of response. Now, during the fear conditioning phase, the next phase, the tone was given with the shock. So the tone and shock were both paired. And after such pairing, the rats started to freeze only after hearing the tone. So why did the rats freeze? And as we had mentioned earlier, the rats froze because they had learned to associate that tone with the shock during the conditioning trials. So that's where the fear learning took place. And that changed something in their neural pathways related to fear. This is why subsequent presentations of only the tone without the shock caused the rats to get into fear state and freeze. So, how is this fear learning taking place? So, how is the brain involved? So, first of all, we have to understand the brain is a very complex organ responsible for thinking, emotion, memory, vision, and several processes allowing the body to function. It really is the computer of the body. Now, when we look at specific areas in the brain involved, it's important to note that the brain has an area called the amygdala that is involved in processing and learning fear. So there are a couple pathways involved in this schematic that we can look at that show how fear is processed. And so what ultimately happens in this experiment, if you take a look at the diagram on the right, we talked about the unconditioned stimulus, which is the foot shock, and the conditioned stimulus, which is the tone. Now, when the conditioned stimulus is given, the neural pathway involved is an auditory neuron that goes to the auditory thalamus that then travels to the amygdala, or goes to the auditory cortex and then travels to the amygdala. Now, if this tone is given by itself, all this will happen. This neuron will go to the auditory thalamus and this will go to the amygdala. And this will not cause a subsequent reaction. So after this happens, this will not cause the lateral amygdala to fire enough that it will cause this these next set of pathways involved in freezing, increasing blood pressure, and hormones. So these set of pathways in red are the freeze response and the response that is exhibited when there is fear. So it's that sympathetic nervous system response. So that fight or flight or freeze response. And in this case, it is there are three separate nuclei involved, but we won't focus on. But there, just to look at these, there are down pathways of freezing, increasing in blood pressure, and then a release of hormones that can happen when there is that fear response. Now, if we look at this other pathway with the unconditioned stimulus and the foot shock, this causes pain. So there is some type of pain receptor here, and that senses this foot shock, and that sends a signal downstream to the somatosensory thalamus. So that's an area of the brain that relays feeling. So somatosensory, so any type of feeling, and in this case, it's pain, and that can either go to the somatosensory cortex and the lateral amygdala. Now, what's important to note is that this pathway causes a downstream effect. So this pathway will activate the fear response. It'll activate freezing, an increase in blood pressure, and a surge of hormones. Now, what happens when both of these are paired together is that this pathway, the auditory thalamus to the lateral amygdala, and this pathway, the somatosensory thalamus to the lateral amygdala, they both meet at the same neuron here in the lateral amygdala. So they both meet in the same neuron here in the lateral amygdala. And so when they're both paired together and they both happen at the same time, these pathways are firing at the same time. So once tone is played, it fires, goes to the auditory thalamus, causes the neuron in the lateral amygdala to fire. Now, when shock happens, it causes this neuron to fire, goes to the somatosensory thalamus, and then causes firing, and then again firing in the lateral amygdala. So when they both happen simultaneously, that actually causes this neuron, the synapse between the auditory thalamus and the lateral amygdala, to strengthen. And that's by a law called Hebb's law, which is cells that fire together wire together. And so ultimately, the synapse between the auditory thalamus and the lateral amygdala strengthens. And that's the actual fear learning that we're talking about. So once this neuron strengthens and causes a downstream active, strengthens, and tone is played by itself later on after the conditioning trials, this is a strong enough synapse to cause the downstream effect of freezing blood pressure and hormones. Then it wasn't strong enough earlier before the shock, but once it's paired and the synapse is strengthened, it becomes stronger. And so we'll look at this concept many more times, but this is just a basic intro into the low roads and high roads that we will focus on. And in this case, we're looking at the low road. So this is the amygdala, fear, a lot of what's going on in the subconscious. Now the high roads a lot involves the cortex and dampening responses, but for the purposes of this video, we're looking at learning fear, and that's where the low roads are involved with this Pavlovian conditioning. Now, to give you a better idea, we have two videos. The links are also provided that you can double click on, and they tell you more about fear and give you a good idea of what we are looking at. So I'm going to go ahead and let you click on these links and watch the videos, and then we'll resume back. Okay, so I hope you were able to watch those videos and get an idea about what fear is. And I think a key point here is that firing of the amygdala provides the emotional response of heightened arousal. So that really is showing what is fear. So the key point is firing of the amygdala neurons of greater than 7 hertz in a rodent is found to signal fear. And so what we're going to do now is look at this virtual lab using Colab. So in this activity, you use the Colab notebook titled One Cell Model of the Amygdala to comprehend how the amygdala fires in response to a sound during the baseline state. So for example, when you hear music that you like, that would be like a baseline state. So you can see how the amygdala is firing. Then you will find out how the amygdala signal fear for the same sound when it has been paired with a scary outcome, like shock, and you'll see how it fires differently. So thus you will learn about how fear is learned at the neuronal level in the amygdala. So what's going on with the amygdala neurons and their firing rates. And so for rodents, amygdala neurons fire greater than 7 hertz is a sign of fear in the rodent. It leads to a freezing response, but below that, it's not causing any issues. So later tutorials will really go into the pathway we described earlier about the tone, shock, and associative learning. But for the purposes of this lecture and this experiment, we want you to go ahead and run this collab. And for this, look at these two things: adjust the tone frequency and or weight to get the amygdala neuron to fire at the baseline value of less than 0.2 hertz. So that's once in 5 seconds. And then we want you to adjust the tone frequency and or weight to get the amygdala to signal fear and fire at a value of greater than 7 hertz, which is just 7 times per second. So we have an accompanying video with this actual collab experiment. So go ahead and look at that if you're having trouble understanding or trouble figuring out what the values are, and that will better explain exactly what's going on. Meanwhile, you can go ahead and pause this video as well and try running that experiment. And if you're having issues, you can go ahead and run the other video as well as accompanying information with it. Okay, so to conclude this lecture, we learned about the neural pathways that cause us to fear, and that fear can be both innate and learned. We learned that Pavlovian fear conditioning, what Pavlovian fear conditioning was, and how it is applied in an experiment involving rats using a tone and shock. And we explored the neural pathways in the mammalian brain that are responsible for learning fear and how the amygdala responds to fear. Specifically, we focused on the pathway that goes from sensory neurons to the thalamus and then to the amygdala. So later tutorials will build on this and provide further details, but in this video, we just wanted to give a brief intro demonstrating what it looked like. So why should any of this interest us? So why do we care about the fear circuit? How does it really applicable to us as humans? And so in humans, so let's again quickly review what fear is. So it's that response to immediate threat. It activates the fight or flight response. It can increase heart rate, blood pressure, blood flow, freeze response. And as we saw in rats, the rats froze. Now, in humans, we have a little bit more complicated brains. And so we can have problems in our fear circuit that may be exhibited differently, such as in anxiety, which is an apprehension about a future threat. But when we are anxious, we do have much muscle tension, vigilance, caution, and avoidant, cautionary and avoidant behavior. And this also happens in pathologies such as depression and PTSD. And also in phobias. And so phobias is a persistent, unrealistic, and intense fear of specific stimuli. So that also causes avoidance behavior. So ultimately, in a lot of these disorders, there's something wrong with the fear circuit. And so if we're able to better understand this and better understand what's going on in the brain at a lower level, we may be able to understand how to dampen it and understand how to better treat patients who have these conditions that may not be so easily explained right now. And if we get a better understanding of it, we may be able to treat it in the future. So that's it for this lecture. Hope you all enjoyed it.